So this is what you should have gotten. The sort of RNA-seq curve with this weird sort of sudden change to a straight line. And then an overall AUC of only 0.73. So in general, it doesn't work as well using normalized counts in a Gaussian model. If you have counts, you should be using counts, raw counts in the negative binomial model for monocle. So the next method we're going to look at is MAST. So MAST is based on the zero-inflated negative binomial model. And it uses a hurdle model to test whether there's differential expression, which combines a test of first the zeros, whether the dropout rate is significantly different between the genes, and whether the continuous non-zero values are significantly different between the genes. And this one also uses a linear modeling framework so that you can consider complex models, including batch, as well as your biological groups. And it's based on the normalized counts. So you need the log normalized, the log normalized data for this case. Yeah, so you first have to create these data frames because it has its own objects again that we have to create. So here we create the MAST object, which is very similar to the other objects where you have the matrix and then the column data and the row data. But again, everything is just named slightly differently. And we have to scale the data. So this is going to scale and account for the sequencing depth in our data if we haven't corrected for library size before inputting the data to the model. And this is how we run the test. So here we're testing for the biological condition while controlling for the total number of detected genes, as recommended by the authors of this method. So in we do the log ratio test again, similar to edge R. And get the summary statistics and the p-values from that. So this method, as I mentioned, tests multiple different things. So it tests the dropouts and the non-dropout values separately. And it combines those into a single score, which is called H, which is why you have to grab only those rows called H for the full test out of the output from this method. And then we can do our ROC curve again. And look at the overall AUC, which is 0.83. So this is roughly performing as well as edge R and the Wilcox test. There are also a couple of slower methods that take a long time to run. So I recommend you try running them, but we're not going to run them now because nobody wants to sit here while their computer whirs away for an hour or more trying to run these. So this is a Poisson beta based general linear model based method called BPSC. And see the GLM call here where you have your design matrix and then your reference cells, reference condition, and the entire data set. And because it's so slow, you have the option of running it in parallel, which I highly recommend if you're going to run this method. And then you set that parameter false to make it run a bit faster, but it'll be slightly less accurate. And the last method is SCDE, which is not GLM-based for once. Instead, this uses Bayesian statistics in a zero-inflated negative binomial model. And it tests for a difference in the mean expression while accounting for the zero inflation of the distributions. Oh, and it has, more recent versions have ways of testing for groups of genes, not just a single gene at a time. So if you want to test whether a pathway is differentially expressed between two conditions, you can do that with this method. But this method is extremely slow. So you have to make sure you have an integer count matrix for this method. And then you fit the error model with all these various parameters to try and encourage it to run faster. And then you have to estimate the prior distributions, and then run the differential expression test here, and then you can extract the p-values using this method. And you have to convert these into two-tailed p-values because it actually outputs just a z-score, not an actual p-value. So this is how you convert that z-score into a p-value. And that's it for this section. So I'll give you, yeah, any question? Oh, in terms of area under the curve. So do any of these methods perform better than a jar or a Wilcox test? I've heard different stories from different people. In general, no, they don't. They all perform roughly the same. It's just a question of how long do you want to wait for your method to run, and how fancy do you want to pretend you're actually doing stuff. Sometimes SCD has performed better than the others for me, but on other times not. So it's very dependent on what data set you're using, which method will slightly outperform the other. Overall, in general, they all perform about the same. So if you have higher total counts, so if you sequenced more, then SCDE tends to work better. If you have lower coverage, SCDE is no longer better than anything else. And SCD is also very slow, so it's not practical to run it on more than 1,000 cells, unless you have a massive supercomputer that you can dedicate it to. So we're going to... Someone's done a full comparison?
Yes.
Oh, yeah. So if you can, okay, so therapad dash. Yeah, see if I put the link. So this is a recent comparison that's unpublished of different differential expression methods on single-cell RNA-seq data with good intermediate poignancy. So these are so nodes is another one that's been specifically designed for single-cell RNA-seq. Same with scDD. However, we've also found that scDD performs terribly and takes forever to run, which is why we didn't even bother mentioning it. Nodes is essentially a combination of a unique normalization method and the Wilcox test. And there's a whole bunch of it. So DSeq2 is on here, Monocle. See, the top performing ones are basically AJAR and MAST and Lemma. So the bulk RNA-seq methods actually work perfectly fine or better than the single-cell specific methods.
But other than mostly for distinct populations. So.
Yeah, yeah. Yeah, this is only for comparing discrete groups. This isn't for along pseudotime. As far as I know, there's only one specific pseudotime method, which is called beam, and it's part of monocle. But for any of the GLM methods, you could just use pseudotime as your predictor. Yeah, and you see the Wilcox test here, which is completely simple, performs almost as well as anything else. And it's really not necessary to look at single-cell specific methods. Ooh, Surette performs quite terribly. Something to keep in mind if you're using Surette. So that's on the Etherpad for this course here, if you want to write down the link to that. Because we've also answered a few questions from people watching this course live on YouTube here. Color?
Font size.
Font size. Oh. Should be possible.
No, probably not.
We'll also post the link to this on the course webpage.
Yes. Can you see this link on top? Can everyone see this link? This one? Hello. We received some questions on the YouTube channel and recorded them in this document. So if you can see link on top of this page, you probably want to make a note. But we'll post it on the website anyway. And I added the link to this differential expression comparisons at the bottom of this page. It's on Twitter, so it's not published yet.
Any other questions about differential expression? Oh, okay, we'll move on to projecting cells across different cell types, data sets.
Okay, so can you hear me? Yeah. So I think we've finished with all the classical analysis of single-cell RNA-seq data. So now I will cover a couple of chapters which are quite new. So these methods are not standard yet, and you won't see lots of them currently in the literature. But they start coming up, and because the amount of the publicly available data sets has grown, and also with all these large initiatives like Human Cell Atlas, now there is a need of comparing data sets from different labs and basically trying to find some more insights by comparing them to each other. So here I'm presenting the package called scMap, which is, we developed this package ourselves. And what it does, it has it has two purposes. So one purpose can be, as I just mentioned, when you have two experiments from different sources, different labs, you probably cluster them separately, but those experiments come from the same organism and the same kind of the same biological system. So the cell types that you identified here may be similar to each other. So then you want to see whether the cell types from this experiment can project to the same cell type in another experiment, which will validate your cell labels, let's say. So that's one way of using the package. Another way of using the package is that when you already have a large collection of single cell data sets, and then you have a new data set from a new experiment, instead of clustering the data set, you can project it to your large collection and identify the labels based on the similarities between your cells and the reference. So you're not doing clustering here, but you can still assign cell labels to your cells. Yeah? Yeah, so the question is whether we need to apply the same normalization for the new data set as we used in the atlas. In the case of scMap, we don't worry about it because the matrix that we use is internally normalized, so it's similar to correlation. So all the similarities vary between -1 and 1. So it allows you to avoid thinking about normalization. So this is not published yet. This is still under review. So I mean, you should take it with a pinch of salt and try it on your own, see how it works, and let me know if you have any ideas. So how the algorithm is, again, we're going to use single cell experiment class as before. So this class is integrated in the package. And for this exercise, we're going to use two data sets, which are human pancreas data sets, so Muraro and Seger-Stolpe. So these data sets are quite nice. They have similar cell types, so they're both human pancreas. They both find the same cell types, but they are from different labs and different publications. So again, we store cell labels reported in the publications. Now we store it in cell type 1 column of annotation, of cell annotation matrix. So you can load the data sets by running these commands. Then for scMap, we are using feature selection. So the feature selection algorithm here is very similar to M3 drop algorithm that Talula presented in the previous chapters. The only difference is that instead of plotting the Michaelis-Menten curve, here we try to plot everything on linear scale, and we identify the same genes which are on the right-hand side of this plot by basically fitting linear model to this bunch of points, and then looking at the residuals of this linear model. So it's the same principle. We try to find the genes which are on the right side of the curve, but instead of complicated fitting, we just use simple linear model. So when we run this function, so we assume that Muraro dataset is our reference. So we want to find the genes that are the most informative in the Muraro dataset, run this function, and it will write all the results back to the single cell experiment object, saying that these genes are the ones that we're going to use, these genes we will neglect. And then we run the second function, which is already a projecting function. And now we want to project scRNA-seq to Muraro. So you define your projection and your reference in this function. And we write the results back to scRNA-seq because we project scRNA-seq. So here, scMap calculates similarities between the cells in the projection data set and the cell types in the reference data set. We have, as I told you before, we have defined the cell types in cell type 1 column. And this is what we're using here. So when we run this, we will have the results again in the call data slot of our single cell experiment object. And the columns will be called scMap labels and scMap similarities. So scMap can either assign your cell to a cell type, or it can leave your cell unassigned. And it will also show you the similarity measures, which range from -1 to 1. So you have your cell IDs, and each cell is either assigned to some cell type in the Muraro dataset, or it's unassigned. It's left unassigned. We can also plot a Sankey diagram. So Sankey diagram will show you the mapping. On the left-hand side, you have scRNA-seq dataset. On the right-hand side, you have Muraro dataset. And because in scRNA-seq we already knew the cell types, right? We also knew the cell types in Muraro. So we can see after the assignment of scRNA-seq dataset, after projecting scRNA-seq dataset to Muraro, whether we have the same cell types or not. Is it clear what I'm saying, or you're confused now? Because I'm confused myself. So are there any questions? What is not clear? The question is, can you be sure whether the differences are technical or biological between two samples? I think you can never, here it's obviously you will have both, because the experiments come from different labs, and probably they did it under different conditions, right? So you have both biological and technical differences. But we are not looking at the differences in this case. We are trying to find similarities, right? So again, here we have two data sets, right? Imagine that we know cell types only in one of the data sets, this one, and we don't know the cell types here, right? We take every cell of this data set and project it to the cell types of the second data set. And then if it's similar to one of the cell types, we assign this cell type to this cell. So basically we label all the cells in this new experiment, right? Once we label them, if we don't know anything about this experiment, that's it, we stop. But because we knew everything about this experiment before, we can compare those labels to the original labels in the publication. And that's what you see here on this diagram, right? So on one side, you have labels assigned after projection. On the other side, you have labels from the original publication for the same data set. And you can see that most of the assigned new labels are the same as in the original data set, which means that projection worked quite nicely for these two data sets. So with scMap, you can also create a precomputed reference. So if you have a good data set where you identified labels of the cells quite nicely, you can create a reference for this data set, and then people can use this reference and project their new experiments to the reference. A reference is usually much smaller than the original data set, so it makes it easy to share the reference with other people, and also make it very fast to project new experiments to the reference. So if you have, let's say you have 100 data sets with very good labeling of the cells, you can create references from these data sets. You will have 100 references, which are small in size. You can share them, and people can quickly project their new data set to 100 other data sets in a very quick way, and see whether the projection is consistent. Yeah.
So you are creating a sort of average signature for each of the
Yes.
labels. Okay.
So in this case, it's pretty simple. So we take a median expression inside of each cell type. So the reference will be, the dimension of the reference would be all your genes against all the cell types instead of all cells. So you reduce the dimension in cell space. So this is the first version of the tool where we just use the median. Now we're working on the second version where we actually store information about each cell, but still the reference is much smaller than the original data set. So once you have the reference, you can use it for your projection instead of the original data set, right? So here we just use this data frame instead of the single cell experiment class, and we get the same results. Yep. So the question is, here we're storing median values of expression in a cell type. Can we store the distribution of the values in a cell type? I mean, for our purposes, we don't need to do that. You probably can, but then storing distribution is the same probably than storing all the original expression matrix, right? So you need to know all the values. You don't reduce the complexity of your data set by storing the whole distribution, I assume. You can either create a model that can be fit to your expression data, but this will take a lot of time. So I'm not sure if it's meaningful for quick projection of data sets. So we wanted to make it as fast as possible so that you can just project very large data sets to other very large data sets in seconds. Any questions on this tool?
If we compare this to cluster, this would be a biased, somewhat biased.
Yes. So if we compare this to clustering, this will be a biased labeling of cells, because you always use a reference as a ground truth. So you believe in your reference. You think that this is true, and then you project it. So for the human cell atlas application, for example, once they have a large collection of data sets where they annotated all the cells quite well, and they believe this is good annotation, and then this can be used for the new coming data sets. For example, because it's if you think about it, if you have 1 million cells in your atlas and you clustered them, then you have another data set with 1,000 cells. You don't want to merge them together and then recluster everything, right? This will take a lot of time. Okay, so the last fancy tool is searching single-cell RNA-seq data. It's also very new. It's even newer than the previous one. So here, the idea is if you have either a reference database with lots of data sets, or you have just a simple, you either have a reference database with cell types, or you have a reference database with cells, and you want to search this database by gene names. So let's say you identified some list of genes in another experiment, and you would like to search your collection of single-cell data by these genes. Find out where these genes are expressed the most. So if you have millions of cells here, it's not straightforward to work on the total data set. So you want to reduce the complexity of this database so you can quickly search by your gene names. So this is similar to any web search, like Google search, when you provide a text, and it will very quickly give you results back, because the complexity of this whole web database is reduced, and it searches not in the whole web, but it searches in some small index which was created based on the whole web. So the idea here is the same. We would like to create an index which is much smaller in size to be able to quickly search our database. So in this case, there is a function called build cell index. If you run it, it will build a cell index for you from your single cell experiment object again. And now we're going to use the same two data sets, Muraro and Segerstolpe, which are human pancreas. And now I define some marker genes for different cell types, so alpha, beta, gamma, and delta cell types. I took these marker genes from the original publication. So this approach is probably biased, but it's just to show you how it works. So if I define these marker genes, list of marker genes, and then I will search the cell index by this list of marker genes, right? And I want to see where these genes are enriched, in which cell type of this cell index these genes are enriched. And if we look at it, we can see that indeed these alpha cell type markers are enriched in the alpha cell type of the data set. We can do the same for, and also we can see it also gives you back, this is a very good feature, so it gives you back the indices of cells in which those genes were expressed together, right? So if all the genes were expressed in the cell from your gene list, you will get the indices of those cells. So you can see, you can look at the cell level and see whether your genes are expressed or not. Yeah. So the question is whether it's co-expression of the genes or whether it's just. Yeah, so in this case, it's just whether they're expressed or not expressed. And whether they're expressed all together in the cell. And also it gives you the cell type where to which this cell belongs to.
So
Yeah.
this is the equivalent of gene-seq enrichment analysis to expression.
Yes. Yes. Yeah. So we are working on implementing this for gene ontology as well. So you can, instead of providing gene list, you can provide gene ontology terms, and it will also return you something meaningful.
And in terms of the human atlas, do you have the equivalent for trajectories for pseudotype? Do you have an atlas of pseudotype trajectories that you can actually reference to?
Okay, so the question is, do we have an atlas for pseudotime trajectories? I think the answer is no. So we don't have, we still don't have the human atlas itself. It still is being built. So, but for trajectories, it will be even more complicated. So I think there are discussions now, what is a cell type? So people still try to define a cell type, and it's very hard to do. So you can run the same searches for beta, delta, and gamma cells. And we can see, at least for this, that for this data set, we get enrichments in the correct cell types. So we use marker genes of some cell type, and we find that these genes are enriched in this same cell type. Now we can do the exercise 2 is more interesting. So we take the second data set, and we'll search the second data set using marker genes from the first data set. So now we won't be biased by just using one single data set, but we're using genes from one data set and searching another data set. So if we do that, so I think this is the results for alpha cells. So it didn't really work well. So we have some enrichment in alpha cells, but we also have enrichment in other cell types. This is the result for beta cells. So this here we have good enrichment in beta cells. Delta cells work nicely, and gamma cells work nicely as well. So this is you probably don't need to use this method yet, but just to give you an idea what we are working on. And there have been a couple of papers recently on bioRxiv with similar ideas. So people now try to integrate different data sets together to be able to understand more about the data sets. Any questions? No? Yeah.
But I don't need to know the cell type. I could create a reference just based on some unlabeled clusters that I predefine or that are not.
Yeah. So in this case, probably what you're interested in is cell IDs. So when you search by gene list, if you don't have the cell types, you can still it will still return you the cell IDs. So then you can go to the cells and see what happens in those cells and why. Well, you will see that these genes will be expressed, all of them together, but maybe you will find something interesting in those cells. So cell types is just for us to validate the method. Any more questions? Okay, so I think we are almost done. So we have one chapter left about SERAT. So this chapter is, I completely copy-pasted it from SERAT documentation. So I will go through it very quickly. How many of you have used SERAT before? 3 people, really. So anyone is doing 10x experiments? The same people, okay. So SERAT is actually, I think it was developed with 10x in mind, and they have a lot of integration with 10x output data. So once you have your data in some file structure on your disk from 10x experiment, then with Seurat you can easily load these files and work with them immediately. So you don't need to worry about single cell experiment class. In fact, Seurat provides their own class. So once you have your data, it creates an object with a Seurat class. So they have their own class, which is not related to single cell experiment class. But because recently this package has become very popular and you can do all the steps that we covered before just using Seurat, starting from quality control, doing clustering and marker gene analysis, and differential expression analysis. So it has lots of, also in the latest version they included this integration of different data sets, the projection of the data sets. So you might want to try that, but as I said, this is a tutorial from their website. This tutorial was applied on this data set with 2,800 cells. In our case, we're going to run it on a dengue data set, the one that we used for clustering, mouse embryo data set, just for, to reduce the time of computation, because this is a small data set, this is a large data set. And well, also, I think what I wanted to show with this data set, because it's small, we noticed that Seurat is actually not very good. It's not very good for small data sets. So once you have several thousand cells, then it's really good. If it's smaller than 1,000, we would recommend use something else. So I just want to give you an example of small data sets with Seurat. Yeah, so we have this. This is the note. If you have less than 5,000 cells, better use SC3 or something like that instead of Seurat. So here, so before doing this chapter, I actually recommend you to restart your R session, because Seurat, because it's so big, it has so many dependencies. When you load it into your R session, and then you load something else, and then your R session will complain that you have too many DLLs, dynamic libraries. So this is one. That's why we actually put it in a separate chapter, because we couldn't compile it inside the clustering chapter because of this error. So if you restart your R session, then start running these commands. So here we take the dengue data set, because we had it in the single cell experiment format. We extract the counts. So in Seurat you start with the counts. We extract the counts and create a Seurat class object. So there are some parameters here. So I think these two parameters mean that we want to keep all genes expressed in more than 3 cells. So this is mean cells parameter with at least 200 detected genes. So that's, you can change this manually. If you go to the website, there are lots of other parameters you can play with. But I just use the ones that they have in the tutorial. So once you created the object, you can start plotting some nice plots. So here we have 2 plots. So n gene is equivalent of total features in scatter. So this is the number of expressed genes in each cell. And UMIs is equivalent of total counts in scatter. So as I said, the main working data sets for Seurat was always 10x. That's why they have this by default. So they always work with UMIs. But it doesn't mean that dengue data set doesn't have any UMIs. Dengue data set has reads, but it's just in the plots they use UMI notation instead of reads or counts. So this is the same data, but when you plot one of them against another one. So number of total counts in your library against the number of expressed genes. And we can see there is some correlation. So the more genes are expressed, the larger is the library size of your cell. So then there is a filter cells function. So in Skater we just subset the data set. Here you use this function. So we define this threshold of 20 million cells, right? So we say we want to remove all these cells because we think they are maybe outliers. I just took this value randomly. There is no meaning in this. You can choose your own threshold if you want. Then there is a normalization step. So there is a function called normalized data. And I think this is the default method, which is called log normalize, which is exactly the same as CPM normalization. But instead of counts per million, here we have counts per 10,000. So instead of dividing by 1 million, they divided by 10,000. Then you can calculate highly variable genes. So this is similar to what Tallulah was talking about. It's probably looking at the variance for each gene. There are some parameters here which I'm not sure about. You can have a look at the tutorial. Some functions here as well. But basically this will create some additional columns in some data frames inside of the object with the values of variability for each gene. It also creates this kind of plot, which is here it doesn't make any sense, I think. But in the tutorial they have a better plot. So it just, I think it shows you kind of which genes, if the gene is far away from the majority of other genes, then this gene is highly variable. And they also provide gene names. So for this data set, somehow it doesn't plot, the plot is not nice. And so this is the number of highly variable genes that Seurat identified. Okay, now we want to remove the confounders. So basically this tutorial is very similar to what we've done before with Skater and all other packages, but in Seurat you can do it, all in one go. You have a question?
So for the variable genes, it includes variable genes and variable spiking as well, or
Do they have spikings in 10x?
No.
Yeah, so I think Seurat doesn't have, they may have spiking functionality, but I'm not sure because in 10x experiment there are no spikings. Yeah. So here we run, so we want to regress out the effect of the library size effect, right? This is what we say here. We want to scale the data and regress out the library size effect. So the dependency that we had, or like if you plot PCA plot, this, you remember the first principal component always had this library size effect. So we want to regress it out to be able to work with some biological effects afterwards. So we regress it out for quite a long time. Regress it out, regress it out. Yes, okay, so finished. So no plots for this one, we just regressed it out. Now we want to run PCA. So yeah, so the point of regressing it out, so when we run PCA, we will see some biological picture instead of some technical artifacts. So for this, we run this function, run PCA. We use only highly variable genes when we run PCA. We also wanted to print out the first 5 principal components and the genes associated with these principal components. So this is actually more advanced than scatter. So here you can look at the genes that are associated with each of the principal components. So I think this there is a print PCA function. So for example, if you run PCA analysis long ago, it will be stored inside of the object, and then if you come a couple of months later, you can print this. This is the same that we saw before. So this is V's PCA function. I suppose these are the same genes that are associated with each principal component, sorted by their strengths. So this genes is the most important for principal component 1. This genes is the most important for principal component 2. And this is the PCA plot. So we want to look at PC1 and PC2. Because we haven't clustered anything yet, we have only one color on the plot. There is another function called PC heat map. So in this function, we actually, I'm not sure what we're plotting in this function. So we plot some gene values associated with principal components. But I'm not sure what these values are. So again, you can go to the tutorial and have a look. But they look quite nice, those heat maps. I haven't used the SERAT myself, unfortunately, so I can't. Do you have any ideas what it is? People who use SERAT? Okay. So, but what are these values? Is it just loadings?
I'm pretty sure the x-axis is the cells, and then it's the loading of that principal component for that cell.
Okay.
And then the score for that gene.
So it's just to give, I think, to give you an idea that there is some biological effect in PC1, PC2. So you see that expression of the genes is very different, whereas PC6, for example, and PC5 is quite noisy. So you probably don't need to use those PCs. Instead, you can use the first 4. But also there are some special functions to find significant PCs, actually. This is the next chapter. So here they use this jack-straw function to find the most significant principal components. So we are plotting the first 9 components, and there is this value is a p-value associated with each of the principal components. And we can see that the first 7 principal components are significant, and this one is already not significant. There is another function, PC elbow plot. So it deals with the graph of your matrix, I suppose. And then here you can find out at which principal component you have a significant drop. And then this would be your last significant principal component, and you don't want to use all the rest. Okay, so once you define the principal components, which are the most significant ones, you can cluster the cells based on these principal components. As I said before, the clustering is based on, it's similar to SNN-click, and then also reusing the phenograph approach from CyTOF data. So briefly, these methods embed cells in a graph structure. For example, k-nearest neighbor graph. So the edges drawn between cells with similar gene expression patterns. And then it attempts to partition this graph into highly interconnected quasi-clicks or communities. So this algorithm is actually quite fast, as I mentioned before. That's why scRAD can actually work on a very large number of cells quite quickly. So you can do clustering of very large data sets quickly. So there is a function called find clusters. So we say we want to use significant PCAs for the clusterings from 1 to 8. You have to define resolution parameter, which is from 0, typically between 0.6 and 1.2. So they get good results for 3,000 cells when they use it in this range. So we actually run it with the same parameter. So once the clustering is done, you can print out the clustering results. So it just saves all the parameters that you used in your last run of clustering. So if you use this object a couple of months later, you can see what you've done before. So scRAD find 4 clusters with this number of cells in each cluster. If we run adjusted rand index, it's pretty low, 0.4. So clustering is similar to all the methods that we considered before, actually. So you can also run tsne using scRAD. Again, we want to use PCAs from 1 to 8. And then you can plot your tsne plot, which looks like that. And scRAD found these 4 clusters now. So we have 0, 1, 2, 3. But tsne plot, again, tsne plot is not necessarily showing you the real structure of your data. That's why these clusters look weird on this plot. But as I said, for larger data sets, usually tsne performs better. So clustering scRAD, when scRAD works on larger data sets, clustering is probably better, and then tsne will represent the structure of your data quite well. scRAD can also identify marker genes. So it does it the similar manner as SC3. So here we say we want to find marker genes for cluster 2, and it will compare expression in cluster 2 to all other clusters. And then you can plot those marker genes and see why they were picked up as marker genes, because expression of these genes is higher in cluster 2. Also, you can plot this. I like this kind of plot. So it uses the same tsne graph that you saw before, but also for all the marker genes, it highlights their expression. So you can see why they are marker genes for this specific cluster. So if you have a nice structure of your data, so if you have like distinct clusters, and then you have a marker gene for some cluster, you will see that it will only be expressed in this specific cluster. So this is very useful for presentation purposes. So there is also a function find all markers. This will run everything against everything. Will take longer time, but then you can plot the results using this kind of heat map. This is similar to SC3. So it will show you which genes are more expressed in some specific clusters, and these are marker genes. So we have one exercise where we want you to compare CERAT defined marker genes against SC3 defined marker genes. You don't need to do it. If you want, you can try. I think this is the last chapter with the materials. We have a couple of more chapters, so I think Tallulah can go through this one.
Okay, so this is just to summarize everything we've sort of gone through and our recommendations. Of course, bear in mind that we did design some of the methods, so take it with a grain of salt. And of course, there are always constantly new methods being developed, so this is our recommendations as of today. Previous versions of this course have had different recommendations because these methods are always changing. So you may want to check back at this page in another 6 months or so when there'll be new methods about. So for experimental design, it's very important to avoid confounding your biological and batch effects if at all possible. Unique molecular identifiers are usually a good idea. They're good for reducing noise, but different methods, but you lose splicing information. So if you're not interested in the splicing information, then unique molecular identifiers is probably your best bet, and it's usually better to use longer UMIs and then correcting for sequencing errors than using shorter UMIs. Spike-ins can be useful for quality control and normalizing, but they're not that useful, and you tend to spend a lot of reads on them, so they're only really worth it if you have money to burn on your sequencing, or you work at a sequencing hub and they'll sequence as much as you want for you. For cell number versus read depth, this really depends on what you're looking at. If you just want to find cell types in your data, you don't need very much sequencing depth. 50,000 reads per cell is usually enough to identify cell types. Whereas if you're interested in regulatory networks or detection of specific lowly expressed genes, you need much higher sequencing depth.